Welcome to G Fun Facts Online. The story of human evolution is one we think we know. It's a grand epic of survival against the odds, of our ancestors learning to walk upright, craft tools, and form societies, all while battling predators and the unforgiving forces of nature. We piece this story together from fragments of bone and stone, but what if a key character in this epic has been hiding in plain sight? A character we normally cast as a villain in our modern story, the toxic heavy metal, lead. We think of lead poisoning as a byproduct of industrialization, a problem of pipes, paint, and gasoline. But a groundbreaking international study proposes a startling new narrative, that our lineage has been intermittently battling lead for over 2 million years. And in a bizarre twist of evolutionary fate, this ancient and persistent exposure may have been more than just detrimental. It may have inadvertently spurred crucial genetic adaptations that contributed to the rise of Homo sapiens and the development of our most defining trait, our complex cognitive abilities, including language. Let's dive straight into the evidence, which comes from an ingenious analysis of our ancestors' teeth. The established timeline of lead exposure, that it was a minor issue until ancient mining and a major one after the Industrial Revolution, has been completely shattered by a study in the prestigious journal Science Advances. An international team of researchers conducted a painstaking analysis of 51 fossilized teeth, and this was a treasure trove of dental history. These teeth belonged to a wide range of hominids and great apes, creating a timeline stretching back 2 million years and across Africa, Asia, and Europe. They had teeth from Australopithecus africanus, an early ancestor from South Africa, Paranthropus robustus, a robust contemporary sometimes called nutcracker man for its powerful jaws, early members of our own genus Homo, our famous cousins the Neanderthals, and of course early Homo sapiens. To read the stories hidden in these teeth, the scientists used a dazzlingly sophisticated technique known as high-precision laser ablation geochemistry. Let's break that down. Imagine a fossilized tooth is like a tree, and its growth layers are like tree rings. From the time a hominid was a child, their enamel and dentine grew in microscopic layers, and each layer trapped a chemical snapshot of their environment. By using a high-powered laser to carefully vaporize impossibly small sections of these layers, one by one, the researchers can analyze the resulting plume of material with a mass spectrometer. This tells them exactly which elements the individual was exposed to and when. It's like a microscopic time machine drilling into the past. And the results were unambiguous. In a staggering 73% of these ancient samples, they found clear, recurring chemical signatures of lead. These weren't just trace amounts. They appeared as distinct lead bands, indicating repeated episodes of lead uptake from their environment. This wasn't industrial pollution. This was a natural, persistent feature of their world. So where was all this prehistoric lead coming from? Our ancestors' world was far from a pristine garden. It was geologically active and volatile. Massive volcanic eruptions could have blanketed entire landscapes with lead-laden ash, which would then contaminate the soil, rivers, and lakes for years. Widespread wildfires, a common occurrence in many ancient ecosystems, would do the same, spewing toxic dust into the air. Hominids would have then absorbed this lead through multiple pathways. They might have drunk from contaminated water sources. They could have ingested it through soil that clung to the roots and tubers they dug up for food. And it would have worked its way up the food chain. Plants absorb lead from the soil, herbivores eat the plants, and our omnivorous ancestors ate both. The study even noted fascinating differences in exposure patterns among the species. For instance, the more frequent, lower-level lead lines seen in Australopithecus africanus and early Homo suggest their varied, opportunistic diet led to a constant, gradual bioaccumulation of lead. They were getting a steady, low dose. In contrast, the more acute, intense lead bands in a species like Paranthropus robustus might point to more dramatic exposure events, like living near an active volcano or surviving a massive forest fire. This discovery of ancient, widespread lead exposure immediately raises a critical question. We know today that lead is a powerful neurotoxin. It's particularly devastating to the developing brain. It disrupts the formation of synapses, interferes with neurotransmitters, and can damage the myelin sheath that insulates our neurons, effectively slowing down our mental processing speed. Even at low levels, it can impair cognitive function, lower IQ, and contribute to behavioral problems. For an early hominid, whose entire survival strategy was based on being clever, on making better tools, on cooperating in a complex social group, on outsmarting prey and predators, such effects would have been a significant evolutionary pressure. It creates a brutal filter. In an environment laced with a neurotoxin, any individual who happened to be even slightly more resistant to its effects would have a better chance of surviving, a better chance of raising healthy offspring, and a better chance of passing on their resilient genes. This is where the story pivots from ancient bones to modern genetics. The research team theorized that this unrelenting environmental stress must have acted as a powerful force of natural selection, favoring any genetic traits that offered protection. Their investigation zoomed in on a particular gene called NOVA1. NOVA1 is what's known as a master regulator gene. Think of it like a foreman on a construction site, directing many other workers, in this case other genes, involved in building a complex structure, the brain and nervous system. And here is the million-dollar clue. Modern humans, Homo sapiens, have a slightly different version of the NOVA1 gene compared to our extinct relatives, the Neanderthals and Denisovans. The difference is almost comically small, a single base pair change in our DNA code, a single letter in a book of 3 billion letters. For years, scientists knew about this tiny genetic divergence, but had no idea what its purpose was. To test the hypothesis that this tiny change was a shield against lead, the scientists embarked on a truly novel experiment. They grew mini brains in a petri dish. Officially called brain organoids, these are remarkable three-dimensional clusters of tissue grown from human stem cells. The stem cells are coaxed into developing into different types of brain cells, which then self-organize to mimic the architecture and activity of a brain in its earliest stages of development. They aren't conscious, but they are an unprecedented window into the fundamental biology of how a brain is built. The team created 2 distinct sets of these organoids. The first set was grown using stem cells edited to have the archaic version of the NOVA1 gene, the one found in Neanderthals. The second set was grown with the modern human NOVA1 variant. Then, they did the crucial experiment. They exposed both sets of developing mini-brains to small, environmentally realistic amounts of lead. The results were stunning. The organoids with the archaic, Neanderthal-style NOVA1 gene exhibited marked disruptions in their development and function. Specifically, the lead exposure threw a wrench into the works of neurons expressing another critical gene, FOXP2. You might have heard of FOXP2, often nicknamed the language gene for its vital role in the fine motor control and cognitive pathways necessary for speech and language. In these archaic organoids, the lead created chaos, disrupting genetic pathways essential for neurodevelopment, social behavior, and communication. It was like static on the line for the very genes responsible for building a communicating brain. But the other set of organoids, the ones with the modern human version of NOVA1, were a different story. They were far, far less affected. The genetic shield held. Our tiny, single-letter mutation appeared to confer significant resistance, protecting the developing neural architecture from the worst of the lead's toxic assault. The implications here are simply profound. The researchers propose a powerful new theory. Our unique variant of the NOVA1 gene may have provided a decisive survival advantage. In a world where lead was a persistent environmental hazard, individuals carrying this protective mutation would have been more resilient. Their brains would have been better shielded, allowing for the stable development of more complex cognitive faculties, more nuanced communication, and tighter social bonds, all the things that are considered hallmarks of Homo sapiens. This theory also offers a tantalizing, if speculative, clue to one of paleoanthropology's greatest mysteries. Why did we, Homo sapiens, survive and thrive while our intelligent and adaptable cousins, the Neanderthals, went extinct? Neanderthals were successful for hundreds of thousands of years, but the organoid experiments suggest a potential hidden vulnerability. With their archaic NOVA1 gene, they were likely more susceptible to the brain-damaging effects of the same environmental lead that we were also exposed to. This constant neurological drag, this slight cognitive disadvantage, could have been devastating over the long term. It might have meant slower adaptation to rapid climate change, less efficient group communication during a hunt, or even slightly higher rates of infant mortality due to developmental problems. It may not have been a single knockout blow, but a persistent handicap that, when coupled with other pressures like competition from newly arriving modern humans, could have contributed to their eventual demise. It's a compelling, if chilling, example of evolution in action, where a tiny genetic tweak in response to an environmental poison helped shape the destiny of two human species. Now, in the spirit of good science, it's essential to apply the brakes of skepticism here. As with any groundbreaking research that challenges long-held ideas, these findings have been met with healthy caution. Experts in the field rightly point out that the claims are speculative. The idea that this one gene was the key advantage is what's sometimes called an evolutionary just-so story, a neat narrative that's difficult to definitively prove. The conclusions are based on a limited number of fossil teeth, and while brain organoids are an incredible technology, they are not perfect replicas of a full, living brain interacting with a body and a complex environment over a lifetime. Much more research will be needed. Scientists will need to analyze more fossils, search for this genetic signature in more ancient DNA, and perhaps develop even more complex organoid models that can simulate more aspects of brain function. Nevertheless, this study has undeniably opened a fascinating new door in our understanding of the intricate interplay between our genes and the world we evolved in. And finally, this journey into our deep past circles right back to our present, serving as a stark reminder of the enduring threat of lead. This research highlights a critical public health message. The problem isn't over. The ban on leaded gasoline in many parts of the world was a monumental success. But lead is the ultimate forever chemical. It doesn't break down. It persists in soil, particularly near old busy roads and around buildings with old lead paint. It's still released into the atmosphere from coal combustion. And a growing global crisis is the unsafe, informal recycling of lead-acid batteries, which exposes entire communities, very often the most impoverished and marginalized, to dangerously high levels of this toxic metal. Shockingly, it's estimated that as many as 1 in 3 children worldwide are affected by lead poisoning today. Furthermore, the research touches on the insidious, multi-generational impact of lead through epigenetics. Epigenetics refers to changes that affect how our genes are read without changing the DNA sequence itself. Think of your DNA as a vast library of cookbooks. Epigenetic marks are like sticky notes and bookmarks that tell your cells which recipes to use and which to ignore. Lead exposure can scramble these instructions, causing changes in a process called DNA methylation, which can lead to genes being turned on or off at the wrong time. And the most disturbing part? Some of these epigenetic changes can be heritable. They can be passed down from one generation to the next. This raises the alarming possibility that the neurological or health effects of a grandparent's exposure could still be echoing in the biology of their grandchildren. In conclusion, the revelation that our ancestors were exposed to and shaped by lead for millions of years adds an astonishingly complex and unexpected layer to the human story. A substance we rightly fear as a modern poison may have been an ancient environmental filter, a relentless pressure that helped forge the genetic resilience that underpins our species' most celebrated abilities. It is a toxic thread woven deeply into the fabric of our 2-million-year history, a reminder that the forces that shaped us are often hidden, surprising, and far more intricate than we ever imagined. And it underscores the fact that our relationship with our environment and the evolutionary journey it drives is a story that continues to unfold. Thank you for listening to G Fun Facts Online.